Hey guys, this is Mr. Mahmood, and you're watching Homeostasis Lecture 2. In this lecture, we're going to finish out our discussion of how homeostasis is able to be maintained at the cellular level. The first lecture talked about the basics of the cell membrane and passive transport. In this lecture, we're going to focus in on something called tonicity, and specifically talk about three different types of solutions, how you can identify the solutions and predict what would happen to cells put inside of those solutions. And finally, we're going to talk about the opposite of passive transport, which is what's known as active transport, and how they can also play an important role in helping to maintain this stable internal environment. So if you remember where we left off before, we were talking about basic ideas of passive transport, and we got into the idea of osmosis, which is the diffusion of water. And with the diffusion of water and the understanding of water, we now can talk about tonicity. Tonicity is all about the understanding of water concentrations and water percentages on either side of the cell and determining and predicting what's going to happen to the cell because of it. So you'll look at this scenario here. I have three cells at the top. They're all identical. So the cells themselves are all made of about 80% water, which means it's about 20% solutes. And again, a solute is everything besides the water in a solution. So the solutes are the different dissolved substances, the different solids, and all the different components that are in the cell besides just water. Beaker number 1, if you look at it, is a 70% water solution. Beaker number 2 is an 80% water solution, and beaker number 3 is a 100% water solution. So if I say it's 70% water, that means that the rest of the percentage to equal 100 is made of other stuff. So we're going to call that again dissolved substances, which is basically everything else that's just dissolved and mixed in with the water. So in this case, if it's 70% water, that means we have a 30% dissolved substance solution. Okay. The 80% water would then be a 20% dissolved substance solution, and then the 100% water would be a 0% dissolved substance solution. The one on the right is what we consider distilled water. That's what distilled water is. It's completely free of any other substances. So these are the three solutions that we're going to be working with here. So even though we're starting with the same cell, what's going to happen to the cell is going to change, all based on water movement. So let's take a look. Let's say the first cell were to be dropped into this solution. Now again, this is an 80% water cell. The cell itself is made of 80% water, whereas the solution on the outside is made of 70% water. In order to determine what's going to be happening here, there are a couple of different ways we have to think about it. If we're talking about how the size of the cell is going to change, we're going to think about the water movement. But if we're naming these three solutions, which you need to be able to do, it's all about the solute concentration. So let's pay close attention to that. There's some basic terms, prefixes and suffixes you need to learn. These are common root words. All of you kids in health sciences and medical terminology and things like that, this is right up your alley. There's some basic suffixes we're going to start with. The first suffix I want to get into is tonic, T-O-N-I-C. If I refer to something as tonic, that means I'm talking about the solute concentration. It's concentrated is what the word tonic means. Like if you've ever drinking tonic water is concentrated water. It's concentrated with minerals and carbon dioxide and things like that. So tonic just means concentration, and it's specifically referring to the solute concentration, which are those dissolved substances, everything besides water. So if we're talking about something being tonic, then we're talking about the concentration of the solution, typically, right? Then the prefixes that we're going to get into are either hyper, hypo, or iso. And each one of these means something very specific. Let's start with hyper. If you're really hyperactive, what does that typically mean? That means you're overactive, more than normal, right? If you ever hyperextend something, like if you hyperextend your elbow or your knee, what does that mean? It means you extend it past where it's supposed to go, higher or beyond the angle that it's supposed to be at. So hyper actually means higher, right? Higher or high is what hyper means. So then hypo is the exact opposite. If you have hypothermia, that means you have a very low body temperature. If you're hypoglycemic, that means your blood sugar is really low. Maybe you're diabetic and you're in a situation where you have really low blood sugar. Hypo means lower. So hypo is the opposite of hyper. It means low. So then what do you think iso means? Iso is right in the middle. It's right in between. So iso means equal. All right, so if I combine these together, I'll name all three of the solutions we're going to be dealing with here. So there are three types of solutions you need to be able to predict: hypertonic, hypotonic, and isotonic. And again, now that you know what it's all meaning, then you'll understand the basic breakdown. So let's start with this first solution. Now, again, the solution itself is 30% dissolved substances, 70% water. Inside the cell, it's 80% water, which means it's a 20% dissolved substance. So if it's a 20% dissolved substance inside, compared to a 30% dissolved substance outside, I want to name the solution. So anytime I'm naming the solution, I'm talking about the liquid outside of the cell. In this case, the liquid outside of the cell is 30, the liquid inside of the cell is 20. So that means there is a greater or higher concentration of solute in my solution, higher solute than what's inside the cell. So this is what I call hypertonic solution. It's hypertonic because there's a greater solute concentration, 30% is greater than the 20% that's inside. So there's a higher solute concentration, and therefore this is a hypertonic solution. Now, if you think about it that way, that's how you name it. The best way to think about what's going to happen to the cell is to think about the water. So look at the water percentage. Inside the cell there's 80% water, outside the cell there's 70. Water again goes from where there's a lot to where there's a little, just like everything else. So in this case, the water is going to move in which direction? It's going to move out. It's going to move from 80 to 70. So the water is going to move out of the cell. What do you think that's going to do to the size of the cell? It's going to cause it to shrink. Exactly. So in a hypertonic solution, because there's a greater solute concentration outside, there's actually less water, or a lower water percentage. That makes sense. It should make sense because the two have to equal 100. So if one is higher, that means the other one has to be lower. So if there's a higher solute concentration, there's actually less water outside, which means the cell has a higher amount of water. That water is going to move out of the cell and cause the cell to shrink. So if you ever need to name the solution, you think about the solute concentration. If you ever want to think about what's going to happen to the cell, think about which way water is going to move. Even though some of these other dissolved substances may be moving in and out, it's the movement of water that's going to have the most drastic effect on the cell. So another way to remember this, if this confuses you, just think of it this way. Follow the movement of water and figure out what's going to happen to the cell. So just like a balloon, if a balloon loses water, if you have a water balloon and it loses water, the balloon's going to shrink. The same thing happens here with a cell. If it had 80% water compared to the 70, water is going to move out of the cell. That's going to cause the cell to shrink. So think of it this way. In a hypertonic solution, the cell will always shrink because there's usually more water in the cell and that water has to go out. So in a hypertonic solution, the cell is going to shrink. Let's look at the middle cell. This middle cell has the same 80% water percentage that is inside of the solution. So both the solution and the cell are sitting at a 20% concentration of dissolved substances and an 80% concentration of water. So in this case, which way would I draw my concentration gradient? The first one was an arrow pointing out of the cell in relation to water. And this time, in terms of water, which way would I draw my gradient? Be careful here. They are equal, but remember, molecules never stop moving. So for every molecule that's going to be moving out, you're going to have another water molecule moving in. So this would be a double-sided arrow, an arrow pointing in both directions, because this is what we consider an isotonic solution. So the cell is going to remain the same size because there's equal amounts of water moving in and out because the percentage is already equal. So now let's compare this to the last scenario. The last scenario, again, is an 80% water cell. This time we're dropping it into pure distilled water. This is 100% water, 0% dissolved substances. So in this case, let's think about naming the solution. The solution on the outside has 0%, whereas the solution on the inside again has 20% dissolved substances, since it's 80% water. So this time, 0 on the outside is lower than the 20 that's on the inside. So in terms of naming the solution, this is what we call a hypotonic solution because there's less dissolved substances in the solution than there is inside of the cell. Now think about it in terms of water movement. There's 100% water on the outside, only 80% water on the inside. So which way am I going to draw my concentration gradient this time? Good. This time it would be an arrow pointing into the cell. And what's that going to do to the cell? Think of a balloon or a water balloon. If you add water, what's going to happen to it? It's going to get bigger. Exactly. And depending on the structure and strength of the membrane and how much water pressure is being put in, that cell could actually get to the point where it'll burst. And this is a big deal. So again, in this scenario, because the water is moving in, the cell is going to get bigger. So again, if all of the hypo, if all that confuses you, just think about which way water moves and how that changes the cell size. In this case, in a hypotonic solution, the cell is going to get bigger, because with lower solute concentration, there's a higher water percentage outside. That water is going to move in, make the cell get bigger, just like a balloon. So I remember it by thinking about a hippo. Hippos are big, right? So in a hypotonic solution, hypo like a hippo, it gets big. So a hypotonic solution, hypo like a hippo, gets bigger, the cell gets bigger. Hypertonic solution, if you're really hyperactive, you burn a lot of calories and you shrink, right? That's how I remember it. So in a hypertonic solution, the cell shrinks. In a hypotonic solution, the cell gets bigger. Hypo like a hippo, bigger. Hyper, like you're hyper and you're burning calories, you shrink. And then isotonic solution, it stays the same because the gradient is moving in both directions. Equal amounts of water moving in and out, and there you go. So all of these solutions can be named either by looking at the concentration of the solute, which is the official way to name them, or just by thinking about which way water is going to move and how that's going to affect the cell size. One way or the other, however you prefer to remember it, you do need to be able to distinguish between these three types of solutions. So these are very important within the cell functioning in terms of homeostasis. A good example of that is what can happen to red blood cells. This is all due to something called osmotic pressure. If you remember, osmosis is just diffusion of water. So that movement of water causing a change in the pressure inside is just called osmotic pressure. So if you look at these three pictures, a typical red blood cell looks like what you see in the middle. Right? So typical red blood cells have that basic shape. So now these are all put in different solutions. Red blood cells have about a 92% water amount, which means it's about an 8% dissolved substance within each red blood cell. So the middle scenario would be red blood cells being dropped in a solution that's pretty equal, about 92% water. Now the solution on the left, what do you think? Do you think the amount of water in the solution is higher or lower than the amount of water in the cell? Think about which way the water is moving. Those cells definitely look like they shrank, like they got smaller, right? So because those cells got smaller, that means that water had to go out of the cells, right? So if water had to go out, that means there must have been more water in the cell, higher percentage of water in the cell than there was on the outside. So I would say the solution there on the left had something less than the 92% water. And in order for the cell to reach this balance, or this equilibrium is the term you may get used to hearing, it had to have water exit those cells, and so those cells shrink in the process. And again, this is a hypertonic solution, because with a higher solute percentage outside, there had to be less water, and that's why the cells lost water. So in this case, the cells shrank. Alright, so in a hypertonic solution, the cell shrinks, and it shows how water was lost there. In a hypotonic solution, it's the exact opposite. It looks like they had to fill up with water. So I would say in this case, there was a higher water percentage outside of the cell than there was inside. So I would say these red blood cells were dropped in a solution that was higher than 92% water, maybe even 100% water. And in this case, the water started moving in to those red blood cells. And they'll continue to move in, especially if you're talking about something that's 100% water, because eventually they'll try to reach this equilibrium, at which point they'll become isotonic. But if you have something that's 100% water and the things that are inside of the cell aren't allowed out, and water is the only thing allowed to move, they'll never actually reach a balance. Mathematically, you can get really close to 100% inside of that cell, but because there's always other stuff inside, more and more water is just going to keep adding and filling up that balloon, and eventually it's going to get so full that in this case the cell could burst. And you see that example there, one of those cells looks like it did actually burst. This actually has a name. So in a hypotonic solution, the cell can get so full that it'll actually burst, which is called cytolysis. Cyto means cell, like cytoplasm is cell fluid, and lysis, especially you honors kids should remember, lysis means to break or separate. So in this case, cytolysis means the cell breaking, and that's exactly what's happening. So in a hypotonic solution, cells can get so big that they actually break or burst, and that's called cytolysis. On the opposite side, with that hypertonic solution, the cell shrinks, and the plasma, the fluid that's inside, is going to be released out. So that means the plasma is actually going to separate from itself. So this is actually known as plasmolysis, where the fluid is no longer all in the cell, so a lot of that fluid has now been separated out, and that's what causes the cell to shrink. So that's known as plasmolysis. The plasma is actually going to separate and make the cell shrink. These are terms more directed towards my honors students, but definitely know this. In a hypertonic solution, when the cell shrinks, the plasma is separated, so that's called plasmolysis. In a hypotonic solution, the cell gets so big that it could actually burst. This is called cytolysis, cell breaking or cell bursting. All right, so be able to kind of describe this basic osmotic pressure. Another good example is what we call Turgor pressure. Turgor pressure is water pressure due specifically to the vacuoles that are inside of a plant cell. Inside of every plant cell you have a large vacuole that's responsible for storing water. If there's plenty of water, the vacuoles fill up like a balloon, just like we talked about with red blood cells. If there's not a lot of water, they start to shrink because the water is being used up by the plant for photosynthesis. So if we're looking at a hypertonic solution, like what you see there on the left, water is in high amounts on inside of the cell. That vacuole, the full vacuole, has more percentage of water than what's outside. So that means the water is going to be exiting out of the vacuole and eventually exiting out of the cell. And all that fluid is going to be released out as well. So again, this is plasmolysis, and it's going to be separated out. Now this doesn't really change the size of the cell because the cell wall is pretty structured. It doesn't really change size very well. What it does do is change the strength of that cell wall. Imagine you have a balloon inside of a shoebox, basically. If you were to blow that balloon little by little, blow it up and fill it up, then the more you fill it up, the more pressure it's going to put on the inside of that shoebox. So from the outside, if I tried to press in on the shoebox, I wouldn't really be able to if the balloon was really full. That shoebox would feel really strong from the outside. However, if I release all of that air and all of the pressure drops from the balloon and it's not even touching the shoebox anymore, then that shoebox becomes a lot weaker. And that's basically how your plant cells work. When they're losing water, the vacuoles shrink, plasmolysis occurs, and the strength of the cell walls start to be dropped, and it becomes really weak and really flimsy. Whereas the other side, in a hypotonic solution, because there's water being moved in, the vacuoles are filling up like a balloon, and that's just pushing up against the surface of that cell wall, making the cell wall super, super strong. So in a hypotonic solution, the cell is going to continue to get stronger as more water is getting in. This is a perfect example of a real life scenario. When you guys go to the produce section of the grocery store, what do you notice happening every couple of minutes? Yeah, you hear that thunderstorm sound, right? And then you see the mist starting to get sprayed on the vegetables. Why do you think they do that? It's not about cleaning them. Think about this. Exactly. If water is going into each of these vegetables or each of these plants and it's filling up those vacuoles, it's putting pressure up against the cell wall. What do you think that's going to do in terms of the strength of the produce? Like a cucumber, for example. It's going to stay super hard, super firm, and it'll be fresh, right? So because water is moving in constantly, it's filling up those vacuoles and pushing up against the surface of the cell wall. That pressure pushing up against the surface is because of osmotic pressure. It's due to water, but it has its own name because it's going on in plants. This is called Turgor pressure. So Turgor pressure is the osmotic pressure in plants, and it's filling up. If it's hypotonic, water is going in, filling up, increasing that Turgor pressure, making the vegetable very strong. Each plant cell is very strong. The opposite is happening in a hypertonic solution. The Turgor pressure is dropping because of all that plasmolysis that's happening, the separation of the fluid, and that cell wall is going to become much weaker, much more flimsy. Pickles are a perfect example. Pickles are just basically cucumbers that are soaked in these solutions over a long period of time. High salt concentrations, things like that. So over time the water is released from these vacuoles, and that's why you notice most pickles are really flimsy. You can bend them around and move them around really well. Compared to an isotonic solution where there's an equal amount of water moving in and out, the strength of that cell wall is pretty consistent and stays the same. So this is what we consider Turgor pressure, and again, all of these have to do with movement of molecules. In a hypotonic solution, the cell is going to get bigger. In this case, the vacuole is going to get bigger. In a hypertonic solution, the cell shrinks. In this case, the vacuole shrinks, or plasmolysis occurs. And in an isotonic solution, the cell stays the same because the gradient is moving equally in both directions. Alright, so all of these will give you good examples of what we call passive transport, molecules moving in the direction you would expect them to move. The final discussion is what we're going to call active transport. In active transport, you're actually getting a molecule to go in the direction opposite to its natural movement. because you're going in the opposite direction, this will absolutely require energy. So active transport, again, has 3 rules, and they're the exact opposite of passive transport. 1, active transport does require energy. 2, the molecules are now going to move against the concentration gradient, so against the natural flow that they would normally move in, which means, third, that they're going from a low concentration to a high concentration. Again, this is not normal. Molecules don't naturally do that. That's basically saying you have a big cluster of molecules and then you have one kind of free floating and it's just going to fire itself into the big cluster and bounce around again. That doesn't naturally happen. If it ever did go in that direction, it would just bounce right back because the natural flow would be to bounce away. In this case, if you're forcing something into an area where there's already more of it, that's active transport. And this scenario, what we call a sodium-potassium pump, is something that happens frequently within your cells, because even though there may be more of something on one side, you still want to get some out, or you still want to get some in. So you look at the diagram here. The left side shows you a basic scenario of potassium, which is the K, moving in the normal direction. You see how there are more of these potassium circles, molecules, inside of the cell. So the natural gradient would be for them to move out. So on the left side here, you see a natural diffusion happening of that potassium going from high to low. On the right side, you see the exact opposite scenario. This time it's with sodium. Sodium has a higher amount outside, a higher concentration outside, lower concentration inside. So just natural diffusion is occurring when these sodium ions go from high to low. Natural molecular movement. Now if you look at the sodium-potassium pump that's in the middle, in this case, both the potassium and the sodium are moving against the concentration gradient. And this diagram doesn't actually do it justice. If you really wanted to show this properly, you would show this potassium actually moving into the cell, because that would be the opposite of the natural movement. Because the natural movement would be for it to go out, but in this case you're trying to force the potassium to go in a direction where there's already a lot of it, just because you may need more of it. So potassium has to be forced in, and this actually requires energy called ATP in order to force the potassium to move in that direction. And then conversely, for the sodium, you would be forcing that molecule to go again from low to high, so that arrow would actually be pointing out if I wanted to truly represent active transport occurring here, because there's already a whole lot more sodium on the outside, so the natural movement would be for it to move in. So if you're forcing it to go in the opposite direction, that's what we're really talking about in terms of active transport. So both of these require energy because you're forcing molecules to go from a low concentration to a high concentration. Another example of it here talks about categories of active transport. The 2 examples that you see here are both what we consider endocytosis. Endo means in. So these are 2 examples of how energy is used to force molecules in, and it's usually because they're just so big that there's no way they're going to pass between the phospholipids, and there's also no way they're going to pass in a channel protein. They're so big that they can't even fit into a channel protein. So if I wanted to get something like this into the cell, I'm going to have to put a significant amount of energy to make this work. I'm going to have to manipulate the cell membrane to surround what I'm wanting to come in, fuse on the other side, and then bring it in that way. So there are 2 categories of this process of endocytosis, bringing things in. The largest of the 2 categories is called phagocytosis. In this case, you're taking a large particle, surrounding it by the cell membrane. It's kind of being pushed in and the cell membrane is surrounding it, and then it fuses on the other end, so that now you have this large particle in its own little vacuole, its own little vesicle, that gets broken off of the cell membrane and is allowed in. And the cell membrane, because it fuses at the top, at no point does it break or allow things to move in and out where it's not supposed to. So the internal environment is always maintained. You fuse it here at the top, and then you're able to break it off in between. So you have this large molecule brought in. This is how a lot of single-celled organisms eat. Like some of the protists, like amoeba and paramecium and things like that, they eat through phagocytosis. They surround their food, fuse it on the other side, and bring it in, and then use different chemicals and enzymes to break down chemically their food and use it for nutrients. Pinocytosis is taking molecules that are slightly smaller but still way too big to pass straight through the bilayer or through the channel proteins. This time, in terms of pinocytosis, it's what we consider cell drinking because it surrounds the particles that it wants to bring in, and it also brings in a significant amount of fluid as well from the outside of the cell. So it brings in a lot of the fluid plus those particles that were too big to pass through into the cell in much the same way. So if you're trying to surround one big thing and bring it in, that's called phagocytosis. If you're trying to surround a lot of pretty big things and bring in a lot of fluid with it, that's pinocytosis. Both of these are examples of endocytosis being brought in, but they can also be described in the exact opposite direction. Let's say I had a big waste molecule that was too big to pass through the membrane, but I needed to get rid of it. I'm going to go through this process of exocytosis, where I'll surround it into this vacuole that's made of the exact same components as the cell membrane. So by the time it hits the surface, I'll break off one end and now fuse to the rest of the membrane, and now whatever was inside the vacuole is now outside of the cell. So the same thing can happen. I make a vacuole that's made of the same stuff as the cell membrane so that when I get to the membrane, I fuse it, I break it open, and now this part just releases out, and I'm left with the original structure of the cell membrane. So exo and endocytosis are both examples of active transport. They both require energy because you're forcing molecules that are way too big to go through the membrane one way or the other. So passive transport requires no energy. Active transport definitely requires energy. Whether it's a pump like what you saw with the sodium potassium thing, or larger molecules being brought in or brought out by manipulating the cell membrane in a way you saw here. Alright, so that covers all the basic movements of molecules through the cell membrane, basic idea of passive and active transport, and how a cell is able to maintain a stable environment by making sure to regulate molecules moving in and out. Make sure you know the different types of solutions, make sure you know the different types of transport. Study this, know it well, you'll see this multiple times throughout the year. Alright, that's it. Next time we talk, we'll be talking about much larger examples of maintaining stable internal environments. Until next time, take care.